Wonderful. Alrighty, I think everybody in the waiting room is in. We probably will see a trickle of people coming in and out throughout the morning. But first I want to start by saying good morning and welcoming everyone. Welcome to those who couldn't join us yesterday and welcome back to those who have. We are super excited about the second day of our workshop, of the Clam Restoration Workshop. Yesterday, we heard about lots of great presentations on clam from Bill, clam distribution, and the history of clam fisheries, aquaculture, and restoration by Dr. William Arnold. That was a wonderful overview, lots of good advice and insight for all of us. If anyone is interested in accessing some of the many documents that were shared to the group in the chat box, I know that Bruce Barber from Virtual Producers will be sharing with you guys at some point in the chat box a link to a shared Google Drive folder where you will have access to them. So feel free to take a look whenever he posts something on the chat box if you want access to that. We will, of course, email you guys afterwards with all of that so you can have it, you can have track of those. Yesterday, we also heard about different groups performing or planning to do restoration projects using clams in Florida, which was really great. Today is just going to be as interesting, but the day will be structured a little bit different. So I just want to take a moment to go over that. We're gonna have our main presentations by the speakers today, who will be digging into some of the ecosystem services provided by the clams and examine what we know about them and what is there still to find out, any advice they can give us. After all the three presentations I will have today, we're gonna take a short break and then we're gonna come back for group discussions. I will go over more in detail about how this will work, but in the general gist is that we will split the entire group into smaller groups and ask you to discuss all of the topics that were presented today with respect to clam restoration. And this is gonna give everybody a chance to make this meeting a bit more interactive because I know it's quite challenging to have everybody talking at the same time. So we're hoping that this will overcome some of that and give chance to people to really dig in a bit more and provide ideas and your thoughts on this. Afterwards, we will reconvene and do very quick lightning rounds on the major points discussed. I will call a representative from each group to just give a very quick main points. Depending on the number of groups, we might not be able to reach everyone because of time, but we'll try to make the best out of this. Quick reminder of housekeeping items. We are working with virtual producers who will be monitoring that everything works appropriately throughout the meeting. So if you encounter any tech issues, please feel free to message them directly. They're in the audience on their virtual producers, or you can message me, especially if there's something going on with your group, breakout group or something funky going on, we can help you fix any issues. Please remember to maintain yourselves on mute during the presentations. The chat box is going to be active throughout the entire meeting, just like it was yesterday. Feel free to add any interesting thoughts or ideas or questions that you might get from the presentations. We will keep track of the questions and make sure that during the Q&A, those 10 minutes, I will go and ask each question. So hopefully we'll have the speakers answer those. And also if anybody in the audience wants to try to answer the question, they're welcome to do that as well. And if we don't get to all the questions, I know that the speakers will probably take a look at the chat box later and try to answer the best they can. Alrighty, I think that is all. I think we had a question about the recordings. The entire workshop is going to be recorded, I think, except for each of the group discussions. But we will definitely share the recordings after the workshop has been is concluded. So with that, I'm going to stop here. And I am going to introduce our first speaker. So let me go ahead and share my screen. So our first speaker of the day is Dr. Bruce Barber. And Bruce is the former executive director of the Gulf Shellfish Institute, GSI. He has a bachelor's degree in biological sciences from Ohio State University and a PhD in marine science from the University of South Florida. Prior to joining GSI, he served on the faculties of Acker College, the University of Maine, where he was the director of aquaculture research, the College of William and Mary Virginia Institute of Marine Science, and Rutgers University Haskin Shellfish Research Laboratory. His research has focused on the reproductive physiology and pathology of commercially important marine bivalves, such as clams, oysters, scallops, and mussels. He has also taught numerous courses in aquaculture, marine invertebrate biology, and advised undergraduate and graduate students. Bruce has published over 50 peer-reviewed articles and book chapters, and he has also served on the boards of the Maine Aquaculture Innovation Center, the Northeast Regional Aquaculture Center, on review panels for NOAA's Sea Grant Program, and participated in the US-Japan Natural Resources panel on aquaculture. Bruce and his wife currently reside in Palmetto, Florida. With that, please help me welcome Dr. Bruce Barber to share his knowledge on water filtration by Hara clams.
Thank you, Denora. Can everyone hear me?
We can.
Great. Okay. Well, again, good morning, everyone. I sincerely want to thank all of you for participating in this workshop and helping us to make it better and taking the time to join us today with your participation. I wanted to talk about water filtration because when any of us think about the ecological services provided by marine bivalves, water filtration is probably the first thing that comes to mind. But in my investigation into this topic over the years, I've found that we really don't know as much about it as I think we need to. So the quantification of water filtration, particularly with respect to clams, is still an open question. So today, I wanted to cover some of the basic aspects of water filtration in marine bivalves, focusing primarily on the quahog or the hard clam Mercenaria. Now, I know that we have a pretty wide variety of background and experience in our audience today, so I thought I'd start with some of the basics. I'm going to start by defining exactly what we mean by water filtration. Then I want to describe the process for how marine bivalves physiologically accomplish water filtration. And this then will lead to the ecological services that are related directly to water filtration. I want to also talk a little bit about how we measure it. Think about how you might measure a filtration rate in a bivalve. It's not as easy as you might think. And there are multiple factors that affect this filtration activity. So I'll talk about some of those as well. And then I'll do a quick review of the literature with respect to water filtration, what we know about it for Mercenaria. A surprisingly small amount of information actually exists. So I'll go over that. And then once we find out where we stand with the terms of what we actually know about water filtration, I want to then ask the question whether we're in a position to be able to apply what we know to a quantification of these ecological services with respect to clams in, for example, a large-scale restoration project. I'm going to highlight a project that the Gulf Shellfish Institute undertook recently to illustrate my points there. And then in conclusion, review the gaps in the knowledge that exist with respect to water filtration. And again, this is what I think we need to understand to be in a better position to adequately quantify the ecological services.
Previous Senates, or how the US Senate, constituted at any one time, has engaged with this.
Okay, so what is water filtration? As all of you recall from your invertebrate biology class, I'm sure you're aware that Bivalves are a class within the phylum Mollusca. And there's a wide variety of bivalves throughout the world, both freshwater and saltwater. But there's a subclass called the Pteriomorphia that have what are called lamellibranch gills. And these lamellibranch gills essentially are an adaptation, because the gills in these animals actually accomplish two physiological processes. They're expanded in size to accommodate both respiration, gas exchange, as in most aquatic organisms, but also the secondary function of feeding. And this group includes the commercially important species that we're familiar with, such as oysters, clams, scallops, and mussels. They all obtain energy by removing suspended particles from the water column. And we call this process filter feeding. Okay, so how does filter feeding actually work? Each clam, and there's an illustration here, has a pair of gills. So these are bilaterally symmetrical animals. So they have two gills. And what we're looking at here is one of those gills with the mantle tissue being removed from this clam lying on its side and exposing the large gills underneath. Each gill is divided into what are called two demibranchs. Each demibranch is then folded to create what are called lamella, or sheets of tissue. And again, this is the adaptation to increase surface area to aid in the filtration process. These lamellae are formed upon closer inspection by thousands of adjacent gill filaments, side by side. And if we take a closer look at two adjacent filaments, which are shown here, we notice that each filament has associated with it three different types of cilia. And it's these cilia that actually do all the work involved. The lateral cilia, which are the ones shown here on the side, actually create the water currents that bring water into the mantle cavity of the clam. They're the inhalant siphon, and then down through the spaces between adjacent filaments. All right, so the water currents bring in the food. The food is then trapped by these longer lateral frontal cilia, which trap particles as they move past between the filaments. They then get moved down to the frontal cilia on the front of each of these filaments. They become embedded in mucus and then get transported toward the mouth. So if we go back up to this larger figure, what this is showing is those particles being trapped by the lateral frontal cilia, being moved to the frontal cilia, where they then are moved distally to the base of the demibranchs, and the mucus string is then moved along a food groove toward the mouth. Okay, so the process of filter feeding or water filtration actually involves two linked but independent processes. The pumping of water itself is called ventilation, and the filtering of particles is called clearance, or we will refer to it as filtration. Now, it's been found that most of these bivalves are highly efficient at removing particles. In the case of Mercenaria, they're almost 100% efficient at removing particles between 4 and 7 microns. So if we're measuring filtration rate and we know that retention is 100%, then logically filtration rate is the same as ventilation rate. These bivalves have the ability to chemically differentiate more nutritious particles from less nutritious particles. All right, and as this food string is moved toward the mouth, it's actually sorted so that the more nutritious particles are selectively ingested, while the non-nutritious particles composed probably of things like sediment and detritus, less nutritious, are rejected. And that material typically embedded in mucus is simply released from the mantle cavity, and we call that pseudo feces. The material that is ingested, again, preferentially things like phytoplankton cells, after they pass through the gut, the waste material comes out as feces, and you can see both types of materials here in this image. Pseudo feces might look something like this. Again, this is material that's been filtered but not ingested. There's a little more up here. I think it tends to be very, very more flocculent, whereas the more compact ribbons are the actual feces here. Both of these are biodeposits and represent organic matter that ends up in the sediment. The two main ecological services then that are directly related to filtration have to do with the decrease in the particulate matter, or this material that's ceston that's removed from the water column, which tends to decrease water turbidity, which in turn increases light penetration and can result in improved benthic plant productivity. The second main service is the benthic pelagic coupling aspect, where the organic matter is taken from the water column and in the form of the feces and pseudo feces deposited in the sediment. Some of this becomes buried in the sediment. Some of it may result in increased denitrification, which I'm sure Ashley will talk about in a few minutes. And it's also been shown that this material can help increase seagrass production, which is a topic of the talk by Dr. Peterson tomorrow. So these are the parameters that we ultimately want to maximize and to be able to quantify with respect to any of our future restoration activity. All right, so how might we measure filtration rate? There are two primary approaches that have been utilized. The first is a static approach, where a clam is placed in a vessel of a known volume of water. Particles are added to the container, usually cultured algae of some sort, and then the decline in particle concentration over time is recorded. And the algal cell concentration can be quantified either by hand with a hemocytometer, or more frequently with the use of a Coulter counter or a flow cytometer. And then filtration rate can be calculated as the ratio of volume divided by time times the log of the original concentration of particles divided by the concentration of particles at time t. A couple of disadvantages with this approach. The first is that while the animal is in there filtering, it's also releasing waste products. And the actual concentration of cells is also decreasing over time, and there really isn't much water flow. So all of these are factors that we know affect filtration rate. So there might be some disadvantages to this approach. However, most of those disadvantages can be eliminated by the use of a flow-through system for measuring filtration rate, where the clams are placed in chambers, such as is illustrated here, and they receive flowing water. The particles coming in to the chamber are counted, the water moves past the clam, and then the particles that pass the clam moving out of the chamber are also counted, typically with a Coulter counter, which gives you not only counts of particles, but also helps you differentiate which sizes of particles are being filtered. And filtration rate is calculated as the flow rate times the ratio of the difference between the concentration of particles on the inlet minus the outlet divided by the inlet. And again, the main advantage of this approach is that you can use ambient seawater containing natural food particles. Very few studies have actually attempted to measure filtration rate in Mercenaria. I'm only aware of these 4, and I'll quickly summarize them for you. The first was Hibbert in 1977, who used a flow-through approach with ambient particles. And instead of counting particles, he actually assessed particulate organic carbon removal. Again, on the inlet versus outlet side of the chamber over a time period of 3 to 4 hours. He used 5 clams that ranged in shell length from 43 to 88 millimeters and temperatures between 12 and 25 degrees C. And for all of these, because filtration rate is dependent upon clam size, I've utilized the basic unit of liters filtered per hour per gram dry weight. So I've normalized them all to gram dry weight. All right, so 1 to 2 liters per hour per gram dry weight for Hibbert. Riesgaard in 1988 actually looked at filtration rates in a number of species, one of which was Mercenaria. He used a static approach with cultured algae on a Coulter counter to count the particles, and each of his trials lasted less than 60 minutes. He had a grand total of 6 clams that ranged in size from 0.17 to 2.387 grams dry weight at a temperature that ranged between 27 and 29 degrees C. He came up with a filtration rate of 1.19 liters filtered per hour per gram dry weight. Carla Beals finished a master's thesis in 2004 under the direction of Shirley Baker at the University of Florida. She looked at filtration rate as a function of both cell concentration and temperature using a static system with cultured algae and a flow cytometer for counting cells. Each of her trials utilized between 10 and 19 clams, which ranged in size from 0.74 to 1.01 grams dry weight. And again, the 2 temperatures that she looked at were 20 and 30 degrees. And not surprisingly, depending on the parameters of the trial, filtration rate ranged from less than a half liter to 1.6 liters per hour per gram. More recently, Echevarria et al. In 2012 looked at filtration rate in a number of invertebrate species, one of which was Mercenaria. They used a static approach, cultured algae, and interestingly, one of the species of algae that they utilized was K. brevis, our local red tide organism. They used a Coulter counter, and their trials lasted 4 hours. They used 8 clams, ranging in size from 1.02 to 1.66 grams, and a temperature range of 12 to 23 degrees. An overall filtration rate of 1.52 liters per hour per gram dry weight. And I've got the asterisk there to remind myself that at concentrations of 700 cells per mil of K. brevis, they found did not negatively affect the filtration rate of Mercenaria. Now, I don't want to talk more about red tide because that's another topic unto itself, but this supports anecdotal evidence from clam growers that during a red tide event, clams do continue to grow just fine. The only problem is they can't be harvested. All right, so again, not a lot of work has been done in this area, and not a lot of different conditions have been examined. So what can we conclude? Well, first of all, it's obviously filtration rate is obviously variable depending on temperature and cell concentration. And these are probably maximal rates. Again, if any of you have ever observed a clam or an oyster or any bivalve in captivity, you know that they're not open all the time. They're not constantly pumping water. And you can't get a filtration rate, obviously, if a clam is closed tightly. And the other thing to keep in mind is that these are all laboratory studies conducted under very controlled conditions for very short periods of time, and really very few replicates were involved in this. So based on this very limited amount of data, can we jump to conclusions such as this for populations of clams in the field or as part of a large-scale restoration project? I really don't think so. I don't feel confident or comfortable making that sort of extrapolation at this point in time. And the reason is that there are a number of parameters that we know affect filtration rate. Temperature, obviously, impacts all physiological aspects of invertebrates. And in Tampa Bay, temperature can range from 10 degrees in the winter to 32 degrees in the summer. Salinity, depending on rainfall, can vary from 20 to 35. Food availability depends on nutrient input, again, and rainfall. And then there's the issue of water flow. We have diurnal tides, so the water is constantly moving, and flow rate also affects filtration rate. And really all of these combined come down to determining the activity level, or how much time clams actually spend pumping water and filtering particles. If there's not enough food available, for example, they're not going to expend the energy to filter if there's not enough food there to make it worth their while. A couple of interesting observational studies, Lusanoff in 1939 noted that Mercenaria was active 81% of the time, and Van Winkle et al. In 1976 found that clams were active only 65% of the time. So it's really difficult, I think, for us to take the results of a short-term lab study under a very limited range of environmental parameters and simply multiply that number by 24 hours to get a daily filtration rate. So to illustrate how inadequate all of our knowledge regarding filtration rate is, I want to talk a little bit about a project that the Gulf Shellfish Institute undertook a couple of years ago. And I think this illustrates both, what I hope it illustrates is what I hope we eventually want to be able to do with respect to quantifying ecological services, but it'll also illustrate the limitations that we currently have to deal with in terms of trying to quantify this. So this was a project that we undertook with our partners, Port Manatee, Eckerd College, and the Bay Shellfish Company. And it was in Tampa Bay, and it was funded by the Tampa Bay Environmental Restoration Fund. We took 300,000 hatchery-produced seed clams, Mercenaria campechensis, and initially placed them in nursery bags when they were about 5 millimeters in size. And then once they got large enough, about 10 to 15 millimeters, we moved them under nets, larger nets. So it turned out we needed 8 nets. These were each 15 feet by 40 feet. You can see them here. And this is actually what the clams look like immediately after they were placed under the nets. We then sampled clams regularly, recorded shell length, dry tissue weight, wet tissue weight, and then we'd go back periodically and take counts for survival, quadrant counts to get numbers per meter squared. We also monitored temperature, salinity, total suspended solids, and total chlorophyll. The seed clams were put out February 2017, and we followed this through to August of 2018. So about a year and a half. And at this point in time, the average clam, the shell length was over 60 millimeters. And we thought at that point they're relatively impervious to most predators, so we removed the nets at that point. And that pretty much was the end of the project. But what we wanted to do was based on the estimated number. So we had numbers of living clams over this time period. We had their size or their increase in dry weight and the measured water quality parameters. We wanted to try to quantify the volume of water that these clams filtered, the total suspended solids that they cleared, and the total chlorophyll that they cleared. And this is what the figure looked like for the volume of water filtered. Now, a lot of assumptions with these. So we used the filtration rate of Riesgaard, 1988, and instead of 24 hours per day of filtration, we used 18 hours, which may not be might not be far off, actually, 18 hours. But because Riesgaard's number was conducted at 27 to 29 degrees, obviously that wasn't particularly accurate for us over the winter months. But nonetheless, we came up with a cumulative volume filtered over this time period of 1.84 million cubic meters. Or at the end of the study, 9,224 liters filtered per meter squared per day. So again, the assumptions were the temperature, all right, 27 to 29 degrees, 18 hours a day. So this is probably an overestimate. In terms of total suspended solids removed, again, the cumulative number was calculated to be 19,147 kilograms, or 192 grams removed per meter squared per day at the end. But again, this is based on the filtration rate and the one temperature and the 18 hours a day, and also the assumption that the clams were 100% efficient at removing particles, which again was probably not true. So this is also probably an overestimate. And then finally, with respect to chlorophyll, the number came out to 16.8 kilograms of chlorophyll removed over this time period, 156 milligrams per meter squared per day removed at the end of the study. But again, the same assumptions, which probably means that this was also an overestimate. So my hope is that we're not so much that we're accurate with this, but that we can at least show the trends and the relative amounts that might be involved here, and what I think we ultimately want to be able to do in terms of water filtration and quantifying the ecological services provided by water filtration. So again, a lot of pretty graphs, but probably not particularly accurate. So this leaves us then with the knowledge gaps, or what we need to know to be able to quantify these ecological services. And if there are any students in the audience today looking for a thesis topic, please take your pick from this list. We need to know more about the effect of temperature on filtration rate in clams. We need to know more about the effect of salinity on filtration rate in clams. Also the effect of food availability, or what the standing stock of phytoplankton is, and the effect of flow rate, and ultimately how all these factors affect the amount of time spent filtering throughout the course of a day. And it's going to be very difficult to do this. We can certainly begin to do it with further laboratory experiments, but I think ultimately what we need to do is try to take some of these measurements out in the field. And there have been some attempts to do this with respect to oysters, for example, measuring chlorophyll and particle removal as water moves over an oyster reef, for example. So I can see where we possibly can do some more sophisticated in the field type of experimentation to get at some of these answers. And once we have these better, more reliable estimates of water filtration, then we can better quantify these ecological services, which in turn, and our ultimate goal really should be then to come up with an economic value for these services. Because as was mentioned yesterday, these are taxpayer dollars that fund these clam planning and oyster planning efforts. And we have to be able to demonstrate an impact, and especially an economic impact. For example, a municipality might have to make a choice. Well, is it more cost effective to create an oyster reef, or plant 1 million clams, or build another wastewater treatment facility? So these are where I think this is where we ultimately want to go with respect to water filtration and probably all these other ecological services that we're going to be talking about today and tomorrow. That concludes my formal remarks, but I'd be more than happy to take questions.
All righty. Thank you, Bruce, for definitely letting us know about water filtration of clams and what is there still much to know. So we have a couple of questions from the audience. And first we have Steve Geiger, who asked, I suppose, about the when you were going over the physiology and the whole process of feeding by clams and filter feeding. Is there an upper size limit to particles clears?
Hi, Steve. Yes, I'm sure there is. I'm not sure I know what it is. I would say it's probably in the range of 10 microns. Again, remember, these animals are adapted to natural assemblages of phytoplankton in the water. That's their primary food source. So I think mechanically they'd have trouble handling particles that are much bigger than that.
Gotcha. All righty. We also have another question from Steve Geiger. So I suppose you're he's talking about your project, GSI project on poor manatee. And he asks, so did you estimate mortality during the study?
Yes, good question. That report, by the way, if anyone that's interested, is available on the Gulf Shellfish Institute website, www.gulfshellfishinstitute.org under projects. We did estimate mortality, and of course that was one reason we took the approach of using the bags and the nets, was to minimize predation. I don't have those numbers right in front of me, but I can tell you that under the nets, the survival was quite high. I think it was 87, close to 90% survival under the nets. Occasionally we'd have a boater or something would run through the nets, tear them up. We'd lose a few clams, or crabs would get under the nets, for example, and we lose a few that way. But it was quite successful overall in terms of survival. And interestingly, after we took the nets off, we went back about a year later and did additional counts and found that even a year later, survival was about 63%, I believe. So that's more or less demonstrated that once they got about to 60 millimeter in shell length or higher, there weren't many things out there that could bother them. I think it was mostly the large whelks. And fortunately, they're not particularly quick, or not particularly, they can't really kill and eat a lot of clams very rapidly.
You're on mute.
Dinora, I can't hear you.
Or you are muted still.
I'm so sorry. Yeah, so that I guess answers the second question that Steve asked, which was, have you ever gone back? So essentially you went back and a year later and you measure about 60% or so survival, correct?
Correct. And those clams that are still out there living, they're still filtering water, and hopefully they've reproduced as well. So we're hoping that there might be a mini clam boom out there pretty soon. I won't tell you exactly where it is, though.
All righty. Let's see. We have a question from Jay Leveroni. Bruce, could you please repeat the resulting numbers of the field study? What water filter, particulate, and chlorophyll removal?
Repeat them. How about
Yes.
I just direct him to the project report?
Sure. And also, Jay, we will definitely have the speaker's presentation posted later on as PDF. So you're welcome to go back and look at the slides if you wish to.
Well, and actually one more thing about that project, which I didn't mention because well, in a way it indirectly had to do with water filtration. One of the most interesting things we found was that the organic content of the sediment was drastically increased by the filtration activity of the clams. So we documented that for sure, which will again lead into the whole discussion of infaunal bivalves and the relationship with seagrasses, which Brad will talk about tomorrow.
Wonderful. We have a couple more questions. Another question from Jay Leveroni. How do clam filtration and clearance rates compare to oysters, scallops, and mussels?
Generally, clams are not as active as scallops or oysters for a given body weight.
All right. Let's see. We have a question from Tyler Heeb. Hi, Bruce. Do you have average gallons per minute, hour, day figures handy?
Well, I can answer that a couple ways, Tyler. Hi. The table that I presented summarizing the literature has those numbers. But I would think if you averaged all those studies, and again, it's hard to do because of the wide range in temperature, your overall average would be about a liter, somewhere between a liter and a liter and a half of water filtered per hour per gram dry weight. And a 50 millimeter clam is about a 1 gram dry weight clam.
All right. We have another question by Nana Riley. Where and how many projects are in Tampa Bay? Or wait, sorry. Where and how many projects are in Tampa Bay? Sorry about that.
I'm not sure I understand that question.
Perhaps he might be referring to maybe GSI projects, I suppose, or I don't know if part of the literature, all the studies that you mentioned were not here, right? They were like using Mercenaria, correct?
Yes, that is correct. All with Mercenaria. I believe Riesgaard did his work up in North Carolina, if I remember correctly. But yeah, none locally. And again, that's part of the challenge. First of all, because Mercenaria and Mercenaria can't beat chances are so closely related, I don't know that there's going to be any difference in filtration rate, but we certainly need to look at it. And the other thing to keep in mind when we got to get a handle on the effect of temperature, we can't just take, excuse me, a bunch of clams and then just drop them into different temperatures and start measuring filtration rate. They have to be acclimatized to the conditions. So a better way to go about that would be to take clams out of the field every month of the year and measure filtration rate in water of that natural temperature. So you get the gradual climatization that occurs in the response to changing temperature on a seasonal basis. And again, this is really the only way we're going to be able to get at this this question of effective temperature and seasonality on filtration rate.
Gotcha. And Bruce, if you don't mind, since we don't have any further questions from the audience, I'm going to ask if Nana Riley would like to unmute yourself and perhaps elaborate on your question in case I might have misinterpreted. Maybe. Nana, if you're there, can you hear us and can you unmute yourself?
Can you hear me now?
Okay, and I'm sorry, I've been probably saying he. I apologize for that.
Molly, hush. My dog is barking.
No worries.
Yeah, I just wondered, like, how many projects has Eckerd done in Tampa Bay? I'm an environmental journalist, and I'm writing an article about clam restoration in Tampa Bay. And I'm just, and clam farming in Tampa Bay. And I'm just wondering, how many projects have they got in Tampa Bay, and where are they? I know he said he doesn't want to say where they are, but a general area, kind of. I'm just curious. Are they North Bay, South Bay?
Okay, yeah, I understand now. Well, we did this particular project was in Lower Tampa Bay, and as far as the Eckerd contribution, they really don't do much. My connection with Eckerd was that they did the sediment analysis for us. I was the shellfish biologist at Eckerd when I was there, but there are other numerous projects involving clams as well as oysters. And I think to get a better handle on that, you could contact Ed Sherwood at the Tampa Bay Estuary Program. Kurt Hemmel, I know with Bay Shellfish, has been involved in a lot of these over the years. Or Steve Geiger at FWC. I think they would have much more information for you on this.
Yeah, I've talked to Ed, but I'll call him again. Thank you.
Okay, thank you.
Wonderful. Well, with that, we're gonna stop the questions and we're gonna move to our next talk. Thank you so much, Bruce, for that presentation. So let me go ahead and share my screen so I can introduce the next speaker, and then Ashley, you will be welcome to share yours. All righty. Our next speaker is Dr. Ashley Smyth. Ashley is an assistant professor in the Soil and Water Sciences Department at UF IFAS Tropical Research and Education Center in Homestead, Florida. Ashley research focuses on how anthropogenic activities impact nutrient cycling in coastal and aquatic ecosystems. Her research centers around where the land meets the water and how activates on land connect to ecosystems in shallow nearshore waters. She has worked extensively on quantifying the nitrogen removal benefits of oyster reefs restoration and shellfish aquaculture. Ashley has a bachelor's in environmental studies and a PhD in marine science from the University of North Carolina at Chapel Hill. Ashley led some of the first measurements of denitrification in oyster reefs and has published 13 peer-reviewed articles on nitrogen dynamics in oyster reefs, oyster aquaculture, and clam aquaculture sites. She has collaborated on projects related to ecosystem services of shellfish in Virginia, North Carolina, Georgia, and Florida. Please help me welcome our next speaker, Dr. Ashley Smyth, who will be talking about nutrient removal and flux by hard clams.
Okay. Thank you for that introduction. I think everyone can see my slides okay?
Yes, we can.
Great. So thank you all for coming today to hear my talk, and also just for being part of this workshop. I'm really excited to take lessons that I've learned from all those other places that I've worked and start thinking about them down here in Florida. And just having this opportunity to share that work, hear what's been going on locally, has been really exciting so far, and I look forward to what's to come the rest of this workshop. There's a lot that builds off of what we've heard in our other talks, and I think that Bruce gave a great job discussing filtration, the importance of feces and pseudo feces, and also that there's just still so many unknowns, and I think that that might be a theme that we'll come back to a lot. So what I'm going to talk about today is work that I did during my postdoc at the Virginia Institute of Marine Science. It was a collaboration with BK Song, Iris Anderson, and Mark Lukenbach. I'm sure you guys, many of you on this call know a lot of those names. And also with Annie Murphy, who was a PhD student on the project at the time. Before we talk about the star of our show, the clams, we have to take a step back and talk about nutrients. Because at the end of the day, I am not a shellfish biologist.
Holly, please.
I'm a biogeochemist, and I care a lot about nitrogen. So nutrient pollution is a global problem. Nutrient pollution is when there's too many nutrients, mainly nitrogen and phosphorus, that end up in our water bodies. Nutrients come from a variety of different sources. They can occur naturally, or they can occur from runoff, where human-related inputs are causing more and more nitrogen to enter these coastal areas. When we get too much nitrogen, or phosphorus, but nitrogen mostly in coastal systems, and that can come from wastewater treatment, runoff, agriculture, etc. That's when we get eutrophication, or we see, as they say here in South Florida, the water is turned guacamole colored green. Eutrophication affects about 65% of estuaries in the United States. It's defined as the increase in rate of supply of organic matter to an ecosystem. I like this definition by Nixon in 1995 to capture what eutrophication is. There's a lot of causes of eutrophication. Those are like excess nutrients, loss of grazers like shellfish, filter feeders, increased organic matter loading to systems, and then also just changes in land use. But the response is then sort of this growth of algae, whether that's phytoplankton in the water column or mats of macroalgae. This is all symptoms then of too much nutrients in our aquatic systems. Eutrophication can look like many different things. Like I said, here it's guacamole green, but for Southwest Florida, I think we're used to seeing the water this more tomato soup red, maybe. This is a photo from the 2018 red tide on Southwest Florida. Red tide is a little difficult to think of with linking it to land-based sources of pollution. It occurs naturally, but then when the algae come near shore, more recent work has shown that the bloom is facilitated and fed by land-based sources of nutrients. So what happens then when we get these large blooms of algae is that as that algae die and it decomposes, we create dead zones. This is where the microbes have sucked out all the oxygen during decomposition. And now we have these low oxygen areas that eventually lead to fish kills, can suffocate any benthic habitat. So that's when we get seagrass die-offs, loss of shellfish, etc. It's really because of this connection between the bloom events and then to nitrogen loading that scientists think that these events, like eutrophication, blooms, dead zones, that they could be mitigated by controlling nitrogen sources and fates within the watershed. Now, shellfish play an important role in eutrophication control because of everything that Bruce just talked about, mainly their filtration and their ability to benthic pelagic couple, so move material from the water column to the sediments. In this figure here, you see that there's this algae in the water column, and then the shellfish will filter that, repackaging it in their biodeposits, their feces and pseudo feces on the sediments. There, these biodeposits are juicy organic matter for microbes and can support microbial reactions, processing of that organic matter. And then that can release nitrogen, ammonium phosphate back up into the water column, or it can even be removed. These transformation rates will depend on the environmental context, so where the shellfish are, the type of shellfish or the type of bivalve. The density is another key factor in the rates of these transformations, and then the microbial community, because at the end, they're in the sediments and they're processing those biodeposits. And because that's really what I think about. To kind of explain this, I put together a little, in more detail, I put together a figure for you. So we have our sources of nitrogen. That's typically the atmosphere from atmospheric deposition, runoff, fertilizer. The nitrogen enters our coastal systems, and there it feeds the algae. The clams in this case will graze the algae, repackage that as their biodeposits, which are organic nitrogen and carbon. And then microbes go to town on this, and they mineralize that, convert it to ammonium, which is a bioavailable form of nitrogen. So that's a form of nitrogen that the clams could use or the algae can use again, assimilation into their biomass. Nitrification is the microbial process that when conditions are right and there's oxygen present, that ammonium turns into nitrate. Nitrate is also another bioavailable form of nitrogen that can flux out of the sediments and be used to support primary production. But nitrate can also be denitrified. Denitrification is the microbially mediated process where that nitrate, that bioavailable form of nitrogen that can be used to fuel the algae, is actually transformed into N2 gas. N2 gas is relatively inert. Only a select group of bacteria can use it. So this represents a complete removal of nitrogen from the ecosystem. So we have the nitrogen that fueled the algae, is repackaged by the clams, and is now converted into N2 gas. So then there's three different fates of nitrogen in shellfish systems. There's that assimilation into biomass for growth. There's removal by denitrification, and also there's regeneration, like the ammonium and nitrate. And then there's burial. Given these three different ways that shellfish can remove nitrogen and improve water quality, we're hearing a lot about this promise of shellfish. We hear that they're an effective method to improve water quality, that they can mitigate HABs through restoration. They remove nutrients, and then there's potential for nutrient trading credits, which we'll hear a lot about tomorrow too. But there's still so much that we don't know. We don't know how these nitrogen cycling differences vary between species, between locations, growing conditions from an aquaculture setting. So I think there's a lot of questions that remain unknown. And we definitely have been focusing a lot on this for oysters. And most of you probably know some of my work on oysters. And more work keeps coming out about the use of shellfish and oysters for water quality benefits. Including this, more recently, we just had this this meta-analysis was just published looking at oyster aquaculture and oyster reefs and their impact on coastal biogeochemistry with a focus on nitrogen cycling. We have this many studies on oysters that we're finally able to do a meta-analysis. The results from this show that oysters enhance removal of nitrogen by stimulating denitrification, that N2 gas production, and it doesn't matter whether they are in aquaculture or in reefs. But what about clams? Less is known about the effects of clams on sediment nitrogen cycling. We often assume that clams will have the same effect that oysters do and that will have the same benefits. But there's a lot of differences between clams and oysters that might not make it the case. For example, clams are infaunal. So oysters are building these reefs on top of the sediments while clams are living in the sediments. And there they can bioturbate. And this can do a lot to oxygen availability and also where their biodeposits end up. They have lower filtration rates compared to oysters and mussels, like Bruce just talked about, and that they're a little bit more sensitive to low quality cestin than some of our other shellfish. So these differences might mean that there could be factors that contribute to whether or not clams remove nitrogen just as efficiently and effectively as oysters do, or are they actually retaining nitrogen? I look mostly at these differences in terms of the fate of nitrate. Nitrate is important because it's an indicator of more runoff nitrogen sources from agriculture and from fertilizer. Ammonium would be more of like the wastewater or human contribution. So nitrate is more of the fertilizer. And there's two different ways that nitrate can be processed by microbes. It can be removed through denitrification, which I just outlined earlier, or it can be retained through DNRA, which is desalinatory nitrate reduction to ammonium. Don't worry, you don't need to remember that. Just remember that denitrification is the removal and DNRA is the retention. And these two processes are competing against each other for that available nitrate. And this is really what the studies that I've done with clam aquaculture have focused on, is what is that fate of nitrate in clam beds? What that fate is will depend on nitrate availability, redox gradients in the sediments, so oxygen availability, accumulation of toxins like sulfide, that rotten egg smell, and then the quality and quantity of carbon, so partially those biodeposits. The majority of my work focused on clam aquaculture was done in Cherrystone Inlet, Virginia. Cherrystone is on the eastern shore of Virginia, and they like to grow clams there. The inlet itself is about 5.7 kilometers squared and is only 1 meter deep. This map here is from a paper by Annie that plots all the different clam leases and clam beds in Cherrystone in black. About a third of the available subtidal bottom in this inlet is occupied by private lease shellfish aquaculture. The watershed in Cherrystone doesn't have any urban impact. It's 40% agriculture and 50% forest. At Cherrystone, it's a large-scale commercial clam aquaculture operation, Cherrystone Inlet. They use hatchery-raised juveniles that are planted directly into the subtidal sediment bottom. And then they cover them with these plastic nets. You can see this image in this image here with this darker color is all the algae attached to the nets. And then here is an aerial photograph showing just the scale of some of these beds in the water. The nets they serve as a great place for macroalgae to attach. And there's a lot of clams in these. They plant clams between 700 and 800 clams per meter squared. What we did for our studies was to focus on the seasonal components of what's happening to that nitrate in these clam beds. We were able to work with the aquaculture company and actually lift up the clam nets so we could sample directly under the net and collect that sediment. So our samples contained both clams and sediment. And then we conducted our denitrification experiments. These have to be done back at the lab, and we used isotope tracers to really make sure that we were able to quantify what that nitrate is doing and where it goes. So here are our results, and this is our denitrification rate. So this is that N2 production. This is that removal. And our results support what people are saying about oysters. Clams have higher denitrification rates than the bare sediment, but not in May. In our May site, we didn't see that increase. In fact, we saw a 9% decrease, but it's not significantly different. In July, we saw an increase in denitrification with the clam, but not over the bare, but it was not significant. The only time we saw a significant increase was in November. I think what's happening in May is that there's the bare sediments were actually affected by pulses of organic matter from the clam beds. At Cherrystone, they use like an underwater lawnmower to sweep away all of that macroalgae that attaches to the nets. And then they let that macroalgae float around in the water. So if they recently had just done that, we would start to see some of those effects in our results. And this idea that the clams are sort of expanding their footprint to that bare site is supported by some of the other data that we have on sediment organic matter and on pore water nutrients. So in addition to denitrification, we looked at that DNRA, so that retention, that ammonium production. And producing ammonium is important because we need ammonium, but too much of it will just continue to fuel that algae growth. So what we found with our ammonium production and retention was that clams increased retention significantly over bare sediment in every single month. Now you're probably looking at this figure and are like, okay, I don't think that your scales are the same, and you would be right. So I plotted those together here. On our left, we have the denitrification rates, and on our right, we have our DNRA rates. So the removal is in orange and retention is in green. Sorry if I didn't say this before, but the clams are the hashed lines and the bare site is solid. I think this figure kind of captures this idea that you do see that increase in denitrification with the clam over the bare, but that you're seeing much more ammonium production and regeneration than you are removal at these sites. This is not what we see with oysters. With oysters, we see very low DNRA, so retention compared to denitrification. More so at oyster aquaculture sites, but less so at restoration, and not nearly of this scale. So what's happening? Why do clam beds in cherrystone favor nitrogen retention over removal? To answer this question, we have to think back to our controls. But let's first talk about what would happen under normal conditions. So we would expect that there is the algae in the water, it's repackaged to the biodeposits, and then we have this nice area that oxygen can get to, and that's when we have nitrification occurring. So that ammonium is then turned into nitrate. There's also nitrate that's in the water, and that nitrate can diffuse into the sediments and deeper into the sediments. And when it gets into that anoxic zone, then we have denitrification. So that removal, that production of N2 gas. However, once that gets down there too, that was when we would have DNRA happen. And that's when that competition can kick on. So at cherrystone, the first control, well, in general, the controls would be the water column nitrate concentration. And in cherrystone, we have very little water column nitrate. Nitrate is really important for denitrification. I mean, it's important for both DNRA and denitrification, but DNRA is more competitive when there's low nitrate around. So those DNRA bugs will just go to town when the denitrifiers are like, no, I can't work yet. So that's a big factor. The other factor is this redox gradient and sulfide production. At cherrystone, our redox interface was was not very deep. So we actually had a lot more sulfidic rotten egg smell than we do at some of our other sites. And when you get sulfide accumulation, it kind of stops some of the biogeochemistry. In fact, it will prevent nitrification. And when we have no nitrification, we have no nitrate. So we have no nitrate in the water, we have no nitrate in the sediments. And then we also have this carbon quality issue, where the clam beds, the clam biodeposits are, because of that net, they're all just sitting in there, and the biodeposits are just getting chomped on as good quality carbon. So all of those conditions, good quality carbon, low nitrate, lead to this retention rather than removal. So why is cherrystone recycling then? It's nitrate limited system, so there's low nitrate availability. There's this continuous supply of labile organic matter. And then we have this sulfide buildup under the nets. And I think a lot of this comes down to the cultivation practices with these predator exclusion nets and this high density of clams. To pull this all together, I took a figure from my friend Annie's work that put this into a basin-wide context for a nitrogen budget to see really the importance. And the white numbers are the megagrams of nitrogen per year from like a budgeting perspective. So they do filter a lot, and you can see that a lot of that nitrogen is actually used for growth. What's key here is that this denitrification number is really small compared to everything else on here. And actually at cherrystone, if you were to think about using the clams for nitrogen removal, you would want to harvest them because you're going to get the largest amount of nitrogen removal through harvest than you would through having them in the sediments and denitrifying, because they're not doing that. They are regenerating ammonium, which actually is then fueling the algal growth and then continuing to support the clams. So in cherrystone, the clams are capable of grazing 100% of that internal recycling. So they basically create this own little closed loop, and they're self-sustaining in a way. So to some take-home messages that I'm sure and questions that you guys are thinking about, is bivalve culture a solution to deteriorating water quality and eutrophication? I think it depends. It depends on location, it depends on type of culture, density, and other environmental conditions are all factors at play. But we can't just ignore, ammonium regeneration. Do oysters and clams have the same potential to improve water quality? I'm gonna say probably not. Infaunal clams are exposed to low oxygen conditions, high organic matter, and sulfidic conditions, which may actually inhibit nitrogen removal and support that retention and release of ammonium. How do environmental variables affect the ability of bivalves to remove nitrogen? In the systems that we worked at that are low nitrate, high salinity, we start to see sulfide accumulation due to the decomposition of those highly labile organic material, those biodeposits that are all leading to retention. So not all shellfish everywhere are going to remove nitrogen and solve some of our water quality challenges. And it's important to understand how those function in each system so we can better manage the clam restoration, clam aquaculture for nitrogen removal benefits. I think there are ways to do that. So I'm just clamoring for new ideas and discussion about new opportunities and additional work to be done, and really looking forward to getting some data from Florida. So with that, thank you for your attention, and I will be happy to take any questions.
Wonderful. Thank you so much, Ashley, for that great overview and in-depth explanation of how it all works. I'm gonna start with, I have just a few questions from me. I haven't seen a lot of people posting on the chat box, but I will just give them time in the meantime to see if anybody else has any other questions. So that was like super interesting. So a lot of that work that you did, you said it was in Virginia, right?
Yeah.
Nice. Okay. So have you started any work in Florida? And it's like, are you looking still at oysters, or are you thinking also about clams or aquaculture here? How is that working?
Some of my colleagues like to say that I have MIMS will travel. The MIMS is this like fancy instrument that you need to measure N2 production. So I am open for anywhere, anything, anytime people want this information. Leslie and I have been working a little bit at some of her clam leases in Cedar Key. And then we hope to get some more data from other locations too. So we're starting to get a little bit more information from here.
Wonderful. Would you expect, I know that it might change geographic location, but do you have a hypothesis or direction of whether you expect to see more or less compared to the location you worked in Virginia, say in Cedar Key or something like that?
Yeah, I think this is super cool because of the way that clams are grown here compared to the way that clams are grown in Cherrystone. Cherrystone is a huge aquaculture operation. I mean, Virginia is the largest producer of clams. They are willing to sacrifice some individuals, if you will, where my impression and talking to a lot of people on this call is that there's a care and attention that's given to the clams here to like make sure you can maximize the number that you harvest. So I think part of that creates these conditions that don't allow for all that sulfide accumulation. Like you're not having dead clams in the sediments. So I think that we might start to, I would expect that there'd be more denitrification and N2 production removal here than we would see at Cherrystone.
Gotcha. Thank you. Okay, so I have a couple of questions from the audience. Patrick Baker asks, I may have missed it, but how much of the NH3 gets into the atmosphere?
So I don't have a full flow from ammonium to nitrate to denitrification. But the overall percent of denitrification is very little from the budget. We're only looking at about 0.03%. So.
Okay. We have a question by Sydney Williams. Thanks, Ashley. Sorry, I lost it. Regarding Annie's nitrogen budget figure, how was filtration of nitrogen quantified?
Yeah, so she just used filtration rates from the literature, probably what Bruce talked about.
Awesome, okay. We have a question by Jessica Frost from South Florida Water Management District, Ecosystem Section Administrator, excellent presentation. Since your area has highest content in all solid nitrogenous, our area has high nitrogen content in all solid nitrogenous fertilizers and breaks down, oh no, urea, I'm sorry. Since urea has highest nitrogen content in all solid nitrogenous fertilizers and urea breaks down in the soil to give ammonium and ammonium oxidized by bacteria to give nitrate. Have you or others looked into the fate of urea in clam beds?
I don't know of any work that's done that. Maybe some from Italy, but this I think like what I think we're missing sort of from all the shellfish work is this like real flow of the nitrogen to really identify how much nitrogen is in the phytoplankton and how much of that is processed and removed through different processing. So that would need an isotope study and they're expensive. So we'll get there one day.
Gotcha. We have a question by Bruce Barber. Great synopsis, Ashley. Again, we need to be careful about overstating the ecological services for clams until we have information. I guess it was a question and more of a comment. Let's see. We have a question by David Moe. So could seagrass take up some available ammonium while in concert with bivalves?
Yeah, so the movement of that, the regeneration of that ammonium would fuel any primary producer. So in cherrystone, we see large mats of that macroalgae, and that macroalgae is supported by that ammonium. So in a system, yes, I think that seagrass are definitely supported by the ammonium from the bivalves, and I think our talks to.
We have another statement by question by Robert Tunstead. Simplified statement here and wanted to clarify sediment versus soil definitions. Would we not want to call these areas of soil that are I'm not exactly sure what that means. Would we not want to call these areas of soil that are growing clams? You have similar biochemical processes that you have on upland subaerial soils as you have in subaqueous soils. Aren't these soils? I'm trying to get into the soil statement versus sediment and.
Yeah, this is a very geological definition. I generally just go with if they're wet, they're sediment, but I'm not a geologist, so I'm sure that's up for just, there's more details to that.
Gotcha. We have a question by Bradley Peterson. Would you expect nitrogen fluxes to be impacted differently from the calcareous sediments of Florida versus terrigenous ones in Virginia?
Yes. Virginia. Yeah, I think that that's sort of what's like fun about being in Florida is the sediments are so different and just how that factors in.
Wonderful. Okay, we have a comment by Barry Hurd. COVID has interrupted some farm work with farmers this last year in Southwest Florida, for sure. Let's see, we have a question by Jay Leveroni. Assuming our estuarine sediments are relatively less organic than Virginia, are they? Or will this be beneficial to the nitrogen removal potential from clam aquaculture in our region?
I don't know if they are or not. The sediments that we worked on in Virginia were pretty sandy. So they may or may not be different than in terms of organic matter than Virginia. But if they are, I think that this is sort of where that deposition of the organic material really matters. So if you have a high organic sediments and then you add more organic matter to it, you're probably going to see more regeneration than you would denitrification. So if you stand to your soils, sediments, you have that organic matter, you'll probably see more denitrification. There is a study that kind of did this from oyster aquaculture in Maryland that I can share.
Wonderful. Thank you. And then one last question by Brittany Wolf-Bryant. Are there ways that we can reduce the DNRA for clam aquaculture? Should there be best practices associated with aquaculture to reduce the DNRA?
Yeah, I kind of see that as a direction to go, is trying to understand what's happening on the farm to manage for denitrification and not for DNRA. And I think there's ways we could do that. It just probably need a little bit more thought and coordinating with all of these people on the call. So.
Wonderful. Well, thank you so much, Ashley, for your presentation and for answering all these questions. We definitely look forward to having you in the audience.
Thank you.
All righty. So now I will go ahead and introduce our last speaker of today. Let me share my screen briefly. Let's see. Alrighty, so our last speaker is Dr. Patrick Baker. He is an invertebrate zoologist. Patrick studied in the Pacific Northwest and Virginia and got a PhD in marine science from the Virginia Institute of Marine Science, College William and Mary in 1993. Patrick has been at the University of Florida since 1999, where he has studied green mussels, commercial clams, oyster reefs, and offshore sand communities, among others. Bivalve mollusks are his most frequent model taxa, and he also teaches invertebrate zoology at the undergraduate and graduate level at the University of Florida and in alternate summers at the University of Oregon. Please help me welcome Dr. Patrick Baker, who will be talking about carbon sequestration by hard clams. Patrick, when you're ready, you're welcome to share your screen.
Okay, I think I'm screen sharing. I'm just not seeing it yet.
I think you might have to select your presentation, or if it asks you, if you have multiple screens, yeah, just make sure you select your PowerPoint. And
okay, can you see it now?
But we can see the notes one, so I don't know if you can, is there another window where maybe you can choose, let's see. Yeah, you see you have two presentations. Yeah, just like choose maybe the other window there.
What is that? Okay, let's try this again.
Yeah, right now we see your desktop, so probably you just have to navigate
Right
to your PowerPoint. I do see your PowerPoint icon at the very bottom on your, yeah, let's try that and see, and try clicking on slideshow, and let's see if that works.
Okay, do you still see the right screen, or are you still seeing the one with the notes?
Brian, do you have any suggestions on how can we see the full slide?
Absolutely, Patrick, we're seeing the notes right now. So when you hit share screen, I would select not the desktop, but the actual PowerPoint presentation. in the item that pops up.
Okay. That, the problem is that didn't actually pop up
Patrick.
this time. Yes.
Hey, Patrick, do you see your display settings? If you click display settings up there above your slide, the down arrow in the display settings part, move your arrow. There you go. And keep going up a little bit more to the left of end slideshow.
It's a little bit above where you have your mouse now, like your cursor.
I'm at the very top of my screen.
Huh.
This was working fine earlier.
Yeah, I know.
I'm going to request remote control over your desktop.
Okay.
All right, great. Can you, Patrick, please head back into your presentation?
Okay. Like that. You seeing the right thing now?
No, we are not.
Okay, let me try.
One more time.
Brian, perhaps we can stop sharing and share again his screen. I don't know if that will help at all.
Exactly, Patrick. You'll have to restart the share again because I did stop that.
Okay. Okay, this is not showing. This is not showing my computer. That's a problem.
On Zoom, go back and click on share screen button again.
Right. And I do not get my computer. I'm getting a bunch of programs, but I'm not getting my computer all of a sudden.
Are you able to select the PowerPoint presentation?
No, it's not there at all. I'm seeing a Dropbox, Microsoft OneDrive. I mean, you can probably see what I'm seeing.
Actually,
no, because you're not sharing your screen at the moment. Again, are you hitting the green share screen button?
Yes. Yep. Maybe I should leave and re-enter.
Brian, what do you suggest?
What I suggest is yeah, once you hit the green share screen button, you won't see. Oh, I know what happened. You're looking at files. We don't want. Okay, so the window that pops up when you hit share screen, up the top, there are 3 different tabs:
right.
Basic, Advanced, and Files. Let's make sure the one that says Basic is highlighted.
Okay.
And selected. And then after that, you should see screen 1, screen 2,
okay.
And then select other applications.
How about that? I selected screen 2, is that right?
You were still not sharing. Did you click share once it's selected? We have to complete the process. There we go. There we go.
Okay. All right.
Here.
Wonderful.
Wonderful.
Okay. Sorry about that. Okay. This presentation is based on a project I did with Dr. Shirley Baker, also at the University of Florida, several years ago. And our paper based on this is cited at the end. Most carbon dioxide in the atmosphere eventually ends up as carbonate rock produced by living organisms, primarily plankton. Corals are second, and benthic bivalve mollusks are a distant third. But there's a couple of points about this reaction that I want to make. First of all, in this middle reaction, notice that the process of dissolving carbon dioxide in the atmosphere is what drives ocean acidification. And the other point I want to make down in the bottom reaction is that the reaction, the standard reaction, the one that we most commonly talk about, in which bicarbonate is combined with calcium ions to make calcium carbonate, fixes one carbon into a rock, into a mineral, but it also releases one as carbon dioxide gas. So one carbon dioxide is released for each carbon mineralized. And that's why I'm calling this talk carbon mineralization rather than carbon fixation. Now, this released carbon dioxide goes back into the atmosphere, and then it cycles back into the ocean. And again, half of it gets turned into a mineral by calcification, and then half goes back. And so eventually, over time, this will end up fixing most of the atmospheric carbon. The problem is the time. This happens on a geological timescale. So in the short term, this is not a means of carbon fixation. This is just carbon mineralization with an equal amount of carbon release, which I'm calling efflux. So I studied this in the Florida cultured clam industry, and I won't go much into that, but just the high points that are necessary for understanding this. We studied the Mercenaria, and we worked on culture sites that were designated and leased from the state with the using commercial culture sites on farms. And in the process of producing clams, it's very similar in some regards to horticulture because the farmers buy what they call seed from commercial hatcheries, and then they lay them out on the bottom where the clams, of course, they don't photosynthesize, but they do feed on photosynthetic productivity. And then there's a grow out in the field, which is key to what I'm talking about. That can be as short as 9 months, or it can take as long as 2 years. It's generally in between that. This is also key. Most of the techniques involve protection under or within mesh netting. And I'm going to be talking about that mesh netting later on. And this website has more information about the industry. So at Cedar Key, where I did most of my work, when I was doing this anyway, they used a two-stage grow out, starting with the purchased clam seed. And they would grow them in these large bags here. This is a used bag. They're 4 feet or 1.2 meters on a side. And the standard mesh is about 5 millimeters. They use smaller mesh for the seed clam, the initial seed clams, but most of the time is spent in these larger clam bags. And most of the clam bags on the lease are these larger clam bags. A fully utilized lease has about 3,200 bags per lease, which is 2 acres or 0.8 of a hectare. And this lease, it shows space in between the clam bags, but according to the clam growers I was talking to who were growing their clams subtidally, they cover every square inch of the bottom. with the clam bags. After usually in the samples I looked at, 12 to 18 months, the clams were brought back. And this is why clam farmers have very strong farm arms. They would do about 60 bags a day. And they'd briefly rinse them on the boat and then bring them back still inside the bags and then sort them. And if everything went well, they'd get between 750 and 1,000 market clams, which in my samples are about 25 millimeters in shell length. The bag also contains the rejected clams, that is clams that were misshapen or had oysters growing on them, dead clam shells, and other species growing in or on the clam bag. And what I wanted to do was estimate how much calcium carbonate was produced as a result of this aquaculture. So that did not include just the clams. So each sample was one clam bag, and I did 36 of these. So I got the calcium carbonate estimate from the harvested clams by counting the clams, measuring a subsample, and then using an equation that we came up with for the shell length and weight. And this was the, at least for these samples, the R square value was very high, close to 1. And then we returned those clams to the farmers. Then we retained the other shell on or in the bag, anything that was saved on a 5 millimeter sieve, which is the same as the clam mesh bag itself. We bleached those, we dried them, and then we weighed them, and then we identified everything to the lowest possible taxon. The bags with the attached oysters, we had to treat a bit separately because the oysters wouldn't come off the bags. And so what we had to do was we bleached and dried and weighed everything, and then we acid washed the bags, which dissolved the oyster shell, and then we dried and re-weighed, and the difference was the weight of the shell. This is a picture of a bag, and you can see some of the common species growing on it. A couple species of oyster. Oysters made up most of what was going on the outside of bags. There could be a lot of other species, but in weight, they were all quite small. And so here in Florida, we have mostly oysters covering the bags. In some areas, barnacles would be the dominant fouling organisms in the outside. So this is what we got on and in the bags. And I'm glad to say, and the farmers were happy to say, that it was mostly clams. And even the material that was not harvested clams was still mostly clams, either dead shell or the clams that the farmers themselves rejected. And almost everything else was oysters. Even though there were over 3 dozen other species that we identified, shelled species, collectively they made up only less than 1% of the total mass of shell. A lot of specimens, but most of them were either very thin-shelled or very small. This surprised us because when we look at it, we often see these other things. And I think there's a couple things going on there. Partly, it does vary from bag to bag, and sometimes we got more stuff. But also, when we're looking at a lot of things all the same, like clam shells, our eyes automatically go to the things that are different, like snails or pen shells, and then we see those and say, aha, there's a lot of those. Our eyes artificially, our brain artificially enhances things that are different. If we look at just the things that weren't clamshell, we can see there's 2 species of oyster, the commercial eastern oyster, and then the smaller crested oyster. And you can't really tell the eastern oyster and the crested oyster apart if they're both small, unless you look at internal characters. most of what was left was not oysters, were either slipper shells, barnacles, or mussels, and everything else. All the remaining 36 species just amounted to a very small proportion. Here's an example of some of the other species. You can see there's predatory snails, pen shells, art clams. The pen shells were quite large, but they were also very thin shelled, and they had a very high organic content. In fact, if you tried bleaching those, they would just dissolve into little fragments. And so I'd have to take them out before I bleached them. And then a lot of species of small gastropods. Small gastropods were by far the most abundant shells in there, but also so small that a lot of them probably went through the netting. After I measured everything, I used these numbers to calculate the amount of carbon mineralized and effluxed. So I added together the shell material from the harvested clams, the shells in the bags, and shells attached to the bags. And then I multiplied that value by 0.96 to calculate the fraction of shell that is calcium carbonate. And I got that number from CNHS analysis of the dominant species, Mercenaria and Crassostria shells, that was done at the University of Florida. So of those species, most of the shell was calcium carbonate, but there was some organic material. Then I multiplied that second number by 0.12, and that is a fraction of calcium carbonate by weight that is carbon. And then I multiplied that by 0.7, which based on other authors' work is the amount of carbon. Remember that there's one carbon dioxide released for each carbon mineralized. That's the amount that is effluxed as carbon dioxide gas. The remaining 30%, as a rough estimate, ends up as redissolved in the waters by carbonate. And then I had to factor in growing time to get carbon per unit area per year. We had an average of 921 clams per bag, and if you include all the associated shell, that works out to 2.9 grams of carbon per market clam that was mineralized. And that works out to about 10 tons of calcium carbonate per hectare per year, or about 8 tons per lease, or about 1.2 tons of carbon that was mineralized. And across the entire industry, based on industry numbers for the number of clams produced, that was about 534 metric tons of carbon in the year that we did this, mineralized by Florida clam farms. Now, to put this in perspective, in 2007, China produced 880,000 tons of shell, of carbon in shell. If we assume that 70% of the carbon dioxide that was produced as a process of calcification was released as a gas, 734 tons of carbon in that year from those same clams that went back into the atmosphere. And again, to put that in perspective, the best number I could come up with was 444 million tons of carbons from coal-fired plants in 1999 from the southeastern United States, which includes Florida. Now, there are a number of waffle points, and I would like to go over these because some of them are rather interesting. One of them is that clam farming may replace naturally occurring shell mineralization. Now, the leases were deliberately chosen to avoid oyster beds, seagrass beds, commercial clam beds, but that doesn't mean nothing was growing there. For example, angel wing clams sometimes use these areas, and we don't really know how much was produced and how much of this persists as calcium carbonate. It'd be an interesting study to do. Nor do we know that just having clam beds there completely eliminates other shell production. There may be some shell production that continues to go on underneath the clams. Shells may be lost from samples prior to harvest. And we did try to look at this. We used some fine mesh nets. Or we didn't look at samples prior to harvest. That's something that we couldn't look at, but we did look at shells that escaped sampling when we collected. Remember that they were brought onto the boat and sprayed. And some of the small species might have washed out of the bags while they was being collected. So what we did was we took some fine mesh nets into the field and we put the bags into those as soon as they came out of the water and then rinsed them off into the fine mesh net. And we did get a small amount of shell that was washed off or through the net. And that yielded an additional 182 grams of fine material on average per bag. And we added that as a factor to our final numbers. And then different sites and different years will definitely produce different results. And so this is an example of mineralization, not a definitive number for the Florida clam industry. Now, there's also some sources of variation in how much carbon was actually effluxed from these animals as carbon dioxide. So first of all, the fraction of carbon dioxide that dissolves back into seawater is an estimate, and it's very hard to calculate, but we do know that it varies with depth, temperature, and possibly other factors. So there's the greatest efflux from shallow waters and warm waters, which includes Florida. So in very shallow clam beds in the Gulf of Mexico in the summer, efflux may be actually higher than 70%. Now, some of the metabolic carbon that is produced by the animal as part of its regular respiration is used for calcification instead of environmental bicarbonate. And so this carbon dioxide would be retained as a mineral rather than released along with the rest of the respiration. So this would affect the total amount of carbon released by the clam. And this actually has been looked at by other authors. There's references at the end. It varies with animal age, but the maximum is around 30% of the carbon that goes into the shell comes from metabolic carbon. What I don't know, I couldn't find out, was how much carbon dioxide is released by calcification versus respiration. And obviously those numbers would vary because the clam respires all the time, but it does not continually lay down shell. And then finally, some biological mineralization processes use a carbonate ion instead of a bicarbonate ion. So a bicarbonate ion is deprotonated, the extra H is kicked off, and then that reaction does not involve any production of carbon dioxide gas. It just goes straight to calcium carbonate because it can combine directly with a calcium ion. However, the process of going from bicarbonate to carbonate does yield an extra proton, and so would increase acidification. None of these errors in carbon efflux would reverse carbon efflux, but it may change how much carbon dioxide is released from the clams. That's all I had to say about that. This project was funded by a Florida Sea Grant, and my major industry collaborators are Cedar Key Aqua Farms and Southern Cross Aquaculture. And I thank them very much for their very generous assistance during that time. And then I had specialized bags produced at Cedar Key, and Jason Curtis was our collaborator at the University of Florida, conducting shell chemical analyses. And then this final slide are the citations. I'll now take questions.
Wonderful. Thank you, Patrick. So I'll just have a few questions for myself. In the meantime, as we see, I haven't seen any questions from the audience yet. So please feel free to type your questions down and we'll go over them. We have a couple minutes for questions. So I guess my first question would be, have you, I know that you did this work in Cedar Key. Have you conducted some of this work in trying to incorporate some of that possible seasonality in you know, examining the seasonality maybe in the carbon that is occurring, you know, throughout the years? Because I know, I mean, depending on the year and when the aquaculture production, you know, they laid out the bags depending on the size and all that, it varies seasonally as well. Have you looked into any of that in your studies?
Now, this was a fairly small study, 36 bags that took several months to actually do. And so it's mostly done during the growing season. Part of the problem with this project was that once people realized that it's not really a method of carbon sequestration, a lot of the funding agencies lost interest in it. So I would like to do more work on it, but the sort of, I think what I would, if I did additional work, I would show the same sorts of results. It would just vary quantitatively in things like how much carbon was fixed per unit time and what the various associated species were involved. The general processes would not differ. But I would like to dig into some of the details, like some of the sources of error, like how much of the metabolic carbon is sequestered or mineralized relative to the overall mineralization process.
Gotcha. We have a question from the audience by Bruce Barber. It says, hi, Patrick, very interesting work. Do we know enough to say whether clams are net CO2 producers or consumers? So are they producers or consumers of carbon dioxide? Again, I am concerned about statements being made about ecological services provided by clams without adequate data. Can you provide any insights on that?
Clams are very definitely, on the short term, they're very definitely carbon dioxide producers. There's many good reasons to have clams. There's many good reasons to eat clams, but carbon fixation as an ecological service in the short term is not one of them. You can't really say that. If you're willing to wait a couple thousand years, then yes, it's an excellent way to fix carbon, but it just takes a long time for the carbon to cycle out of the atmosphere back into the ocean to where it can be fixed. So as an ecological service, no, I would not say that clams are carbon consumers.
Gotcha. Thank you for the clarification. Bruce mentions. How would you, I guess while we wait for any other questions, I was curious about if you had any thoughts on how coastal acidification may affect this whole like carbon sequestration, remineralization, or the effluxes. Do you have any thoughts on that?
We know that coastal acidification affects clam reproduction. One of the papers I saw on oysters showed that it didn't seem to greatly affect adult oysters, at least at the levels they were looking at. But to get to adult oysters, you have to have oyster larvae. And so, as some of you probably know, some oyster hatcheries in some parts of the world have already had to shut down production because of coastal acidification. And so that is the point right there that's going to slow this down. I do not know enough about geochemistry to know whether acidification will significantly reduce overall calcification by adult clams.
Okay. Perfect. Thank you. I think we do not have any more questions for now from the audience, but if anybody can think of anything else, feel free to add in the chat box, and we'll make the best, try our best to answer your question as we go. Thank you so much, Patrick, for the presentation and for being part of the workshop. Oh wait, we have one more. Understood, but what about relative to other forms of meat production? Would it be better than terrestrial production, pigs, chicken, beef?
Interesting that you mentioned pigs, chicken, and beef. Those are all terrestrial, those are all vertebrates, and so they don't use calcification for the most part. They mineralize calcium phosphate, and so that doesn't involve carbon mineralization. I've been preached many times about how much carbon dioxide is produced by beef production, and you've probably heard that too. Chickens are kind of interesting because the egg industry calcifies an enormous amount of carbon in the form of eggshells. And it'd be interesting to know how much all of that would go back into carbon, all of the carbon dioxide produced as a form of calcification and go back into the atmosphere. And egg production is a much larger source of mineralized calcium carbonate than the shellfish industry is, at least in the United States.
Steve, I don't know if you wanted to add something or clarify about your comment.
Yeah, I can chime in. I mean, I'm not disagreeing with your data. There was a talk at NSA, and I don't know if you were there or not, where you know, we have to eat protein somehow. And so even if as a restoration tool, you're not going to get carbon credits, I still have always felt like there is that that conversation which, you know, you're eating down the food chain, and potentially shellfish are more carbon neutral for the atmosphere than some of the big grazers. I don't know, you know, all the stoichiometry of that. It's just was kind of curious, but you kind of answered that. So
yeah, I think you're right. They eat at the very bottom of the food chain. They eat these primary productivity directly with no intervention by humans. And so we don't transport the food, we don't fertilize the food. It comes straight out of the sea. And so it's almost like, that's why I called it similar to horticulture, because sunlight feeds the food directly that we are feeding to the clams, or that the clams are eating without any intervention by us. So I don't know how boats compare to tractors as far as carbon production. I imagine they'd stand up pretty well, but I would think that if you could persuade everybody to eat clams, you could get more protein per unit area than you could certainly from beef, probably from pigs or chicken.
Wonderful. If there are no other questions, then we thank you, Patrick, for the presentation. And yeah, we're gonna now switch to the rest part of the meeting. We're gonna take a small break, and it is 11:01. We'll come back at 11:11 or 12. So just hang in there, take a break, and we'll come back, reconvene in a moment. All right. And we are back. Hope you had a nice little break and you have enjoyed the presentations for now. So what we're going to do now for the remaining of time is a little bit different, but we're going to split all the participants present into smaller groups of about 10 or so. So the goal of these groups is so that we can share thoughts on the presentations that we had today, and especially ideas of items that we still need to figure out, pretty much discuss gaps and information necessary that will make the point that clams can be used for restoration with respect to the topics that were discussed today. So what we will ask you is that please, in each group, you're going to be self-directed because I cannot be in all the groups, unfortunately. You are going to choose someone who will be taking notes, and this person, of course, welcome to participate in the discussion, but this person is going to be in charge of just writing in a Google document. We will share it with you in that group, all the main points discussed in your group. Take about 10 minutes to discuss each of the topics that were discussed today, like filtration, nutrient removal, carbon sequestration. And well, after that, it will be about 30 minutes. We'll bring you all back and we will ask you guys do a lightning round on the main things that your group discussed. So it has to be really quick because we're running out of time. So those will be like, okay, tell us like the main 3 points that your group talk about or something like that. And so Brian, if you are ready, I think I have already set all the instructions. We will, share with you the document, and Brian is going to go ahead and split us all into groups. So feel free to unmute yourself then and participate in the discussion.
Yep, and as just a reminder, I will be hopping into each breakout room to share the document link that is hosted on Google Drives for our communal note-taking. As if everyone is ready, here we go.
Perfect. All righty, I think we are coming back to the big group. Let's see, I think we still have a few more members coming back. All righty, so we have very little time to recap. So I'm gonna go ahead. I think we had about maybe 6 groups or so. So I'm gonna just call a representative from group number 1. Give us the take-home message that you guys discussed about the different ecosystem services.
I don't remember what number we have.
I think we had number 1.
Oh.
All right, go ahead.
Yeah, so our question was, what are some of the unknowns about clam restoration? And so we came up with several, we discussed several points. We wondered if there was, given the efforts to restore clams by leaving out wild clams, if there is a signal from the very large number of clams in aquaculture farms, especially at Cedar Key, if there's a signal that we can see in recruitment in the surrounding areas. And if not, and we don't have any evidence that there is a large signal, then why isn't there a signal? Why aren't those things reproducing and filling the surrounding area with clams? The second one, we talked a bit about clams, about sediment, and it turns out that Todd Osborne has a bit of data on this that he's willing to share with people, unpublished data, the effects of sediment on optimal clam habitat. And then the third thing we talked about was what sort of effect that harmful algal blooms would have on clam restoration. That was it.
Alrighty, so is that a defect of the algal blooms, like the clams on the algal blooms? Is that like what the unknown?
No, the effects, I think there's effects. Well, I suppose it could go either way. We were just talking, I don't think we really got into that cause and effect.
Thank you so much. Okay, group number 2. I was in group number 2. I'm gonna say, Gary, give us the take-home messages.
I love being volunteered.
Voluntold.
No worries. So a couple of our take-home messages, we were talking about the filtration capacity for clam aquaculture or clam enhancement, thinking about monitoring this in the future, that flow-through systems definitely need to be utilized in these future monitoring experiments to help us create some consensus values. And actually that was something that we went through for our 3 different topics, was to make sure that we're not overselling the individual components for these for aquaculture, for clams and oysters, other shellfish whenever we're doing these systems. Again, more specific targeted experiments. We also talked about nutrient cycling and removal, and brought home from, I think it was Patrick's discussion on the benefits of nitrogen removal may not be fully realized unless we're harvesting the clams, and in a lot of our systems that's a very difficult long-term goal. Only the bottom quarter of lower Tampa Bay is available for harvest right now. We have a couple of aquaculture product companies invested there right now and that are producing, but it'll be a very long reach before we can achieve that. However, some of the other goals that we talked about for clam, for oyster restoration would include, in Tampa Bay, we're looking at it in the old Tampa Bay to help us ameliorate the pyridinium blooms in the summertime, to see if we can get the shellfish to filter feed on some of the larval stages of the pyridinium Bahamense. And we're working with FWRI. I think I just saw Suganda on the call as well, and she's one of the PIs for that project. I've shared a couple of the reports that they did for us over the past couple of years. Also thinking about the pseudo feces and the feces as an important consideration that was discussed during the nitrogen removal process. And then our last thoughts were on carbon sequestration. Jay made the point that shellfish aquaculture, and this is based on talks earlier today, probably not the best tool for mitigating atmospheric carbon dioxide. It's a tiny piece of what shellfish might do, and that that gets to one of our final thoughts was that the shellfish production, it's a multifaceted tool. It is touching on many different things, whether they are important for habitat, for either consumption or protection, for wave attenuation, for filtration activities. But so there's a multi-pronged set of reasons for us to be conserving, restoring, enhancing these systems.
Gary, can I add one final point that I mentioned that I want to make sure that we get across to the larger group?
Absolutely.
And that is that, the biggest concern for many of our estuaries here is nutrient reductions. And certainly aquaculture and shellfish aquaculture and enhancement can contribute to that. But we have to remember that it is part of a larger nutrient management strategy, and it's only one part of it. We are focused primarily on wastewater practices, stormwater practices, and other things, and this can be one of the tools that we consider. There's a lot of NGOs and others that are wanting to do shellfish, oyster gardening and other types of shellfish practices, putting things under their dock and stuff. And we want to encourage it, but it's also an opportunity to say that that's just part of a larger nutrient management strategy that all of us are dealing with. And it's a piece of the puzzle. That's simply it. We don't want to oversell it, but we certainly want to include it in a overall nitrogen management strategy. Thanks.
Wonderful. Group number 3, representative, please give us your take-home message of what you discussed.
I believe we were group 3, but just going to be real brief. I want to give anybody else in the group a chance to chime in. We had a bunch of really good diversity of experience and different sort of objectives for people attending the workshop, so that was cool. We didn't really talk specifically about the 3, goals of restoration, I guess, for today, but sort of broadly, one of the things we did touch on was that there's obviously a lot of unknowns everywhere in terms of the specifics, but water filtration as a benefit of shellfish may be one of the more solid ones and easier for people to understand, for sure, when you're going out and talking to the general public about these sort of things. So that was one of the big takeaways. Anybody else from the group?
I just want to mention about the surveys, the soil surveys. I think one of the pillars or base foundation of any restoration process is the soil health and knowing what we have on what kind of soils we got and what we can do with them or whatnot, and how they change over any human activities that we do in these areas. I think as the USDA and RCS grow in this the subaqueous spreads that we're developing, and as we get more in touch with you all and as we learn more from the local experts, we should be able to produce another tool for your management in the near future. So do not forget about the soils, please.
Alrighty, anything else from group 3? If not, I will move on to group 4. Okay, let's do group 4. Anyone can summarize what you guys talk about?
I'll jump in since I was the one who talked the most in that group. So it is a very complicated whole ecosystem thing to study. But one thing that really we were motivated to talk about was that nutrient reduction. And knowing that, and according to what Ashley presented, great presentations, the harvesting for aquaculture is the one that reduces or removes most of the nitrogen. And we see that. But in the other hand, for restoration projects like what we do in Sarasota Baywatch, is that it's not just the organism, the people who eat the clams that are going to be removing nutrients. Is the predators, the natural predators who be there removing and utilizing those food from the clams to grow and to continue, the crabs, the fish, whatever who comes and eat it. And also, and we will hear that tomorrow, is the nitrogen being absorbed by the plants, which are the seagrasses, who are great enhancement for the area. So we can see these clams as an enhancer for food production, as aquaculture, but also as a preservation or restoration area. And plus they're gonna be reproducing. So that's the other benefit of that, having the clams. So those two alternatives, it's not just the removal by aquaculture, putting it out, but also the nature itself removing the nitrogen by their other components that they have. Anybody else who wants to add something else about that?
Yeah, I'll piggyback off of what Ernesto was saying. I think that a bunch of us stated that the clams in certain estuaries or restoration areas, they're all different and they're all really dynamic. And a lot of these studies that have been presented today and yesterday and probably tomorrow, they've touched on really good things and really good points. And the example used in our discussion was, it's like trying to study the human body, but only looking at your hand, and you don't really see how the rest of the body functions around that hand, meaning, how is like flushing, how is the soil, how are the nutrient loading in that area, what are the predators in that area. So a lot of we need more of a general, broader understanding of each ecosystem and how the clams interact with it, as opposed to just like what the clams are doing in that particular area.
Wonderful. Thank you. All right, now we have any group 5?
Yes, I'll take group 5. So we covered a lot of ground, and these are in no particular order. With regards to the harbor oyster beds, there's not a lot of public support for restoration efforts. There was also a discussion on ways to stabilize salinities and improve water quality. There was also a discussion on stressors, in particular metabolic indices that change really fast. They are more responsive depending on the enzyme that's measured. We also talked about whether or not aquaculture or clam or oyster restoration, or to coin Bill's phrase, enhancement, can be used to stabilize seagrass health. There is a concern about real estate value and waterways, but there is value in increasing light attenuation and commercial harvest. But there's a caveat to that, in that they take up harmful algal blooms and E. coli. We also talked about sedimentation issues. There's, I guess, no commercial fishery, I guess, for clams, if you will, or clam harvest, sorry. And, but they're not coming back. So there's always that question of whether or not it's a water quality problem or if it's a clam problem. And then, there was a comment that Mercenaria campechiensis prefers seagrass beds. So can we use seagrass beds as, a restoration effort? However, there is recognition that you would have to do a lot of manual labor in terms of hand placing, seeding, and whatnot. And it's hard to measure contribution to those habitats. And lastly, funding sources, when the industry isn't as interested or isn't doing well, it's really hard to get funding sources to continue research.
Wonderful. Thank you for that summary of valuable points. I think we have one more group, if I'm not mistaken. Do we have any representative from the last group?
Yeah, this is 6. Yeah, I'll try my best. I did take some notes. So So the question was, please discuss any gaps in information as well as information necessary to make a point that clams can be used for restoration. I'm not sure if we really got specific about gaps. One, I think probably seems like from our discussion, the most important thing is really defining those restoration goals. Often it was cited by a few people in our group that the interest or demand and enthusiasm from stakeholders for shellfish restoration is actually quite high, but that needs to be tempered with the realities of where the habitat is suitable to do restoration, coupled with oftentimes restoration projects or enhancement projects, whatever you want to call them, are limited by where they can be sited due to the regulatory environment. So, or the agencies responsible for permitting these kinds of projects. So defining the restoration goals, determining whether or not the habitat can support that restoration. Someone had mentioned that what had grown there historically may not, the reality is that it may not grow there now, and we need to better understand the current conditions that might limit a particular, whether it's oysters or clams or any other species, from being enhanced in that particular area. The other thing that came out too, I raised a question about, intensive culture of clams. Does that affect wild populations outside of that? Is there any kind of spillover, from those clams reproducing? And Leslie had pointed out in Cedar Key that it's likely there's very high predation rates because they don't find a lot of clams outside of those culture areas. So maybe getting a better understanding of predation rates wherever you're planning those enhancements was, I think Matt covered that a bit yesterday, but it may vary. Of course, it may vary according to where you are too. So there was also comparing oysters and clams. There's there's some pretty good arguments for oyster restoration because they form habitat, and in the case of Southwest Florida, they've lost a lot of reefs as well. So I think that's the main thing, is defining those restoration goals and determining whether where you want to do a project, is it still suitable to, can the area still support that enhancement effort? And if anybody else wants to chime in from our group too, I hope I didn't leave anything out. So.
Thank you, Vincent. Just want to thank everybody for, I think that we only have 6 groups. Just let me know if there's any groups that I missed. But I definitely want to thank everybody for participating. These have been definitely a fruitful conversation. I hope you guys have enjoyed as much as I have. Please, I want to just remind everybody from the groups that we did share a Google Drive document. So please make sure that your notes are transferred there if you haven't, so we can have all these great informations and thoughts that you guys discussed, since we weren't able to be in every single one of those groups. Lots of great remarks. I really don't have much to add other than all the thoughts are valid, and there's still so much work that we need to do and so much science to be done. But this is a great way to start the conversation, and I really identifying those points that we really need to focus more. Before I close, I want to invite anybody who has any last thoughts or remarks that they would like to share with the group. Feel free to speak now. No? Shies? Okay. I will see you tomorrow bright and early again. And you guys have the wonderful rest of the day. And I will see you at 9 o'clock tomorrow.